Okay, so we're on to chapter 18 now. This is skeletal muscle structure and function. What you guys are going to notice here is this section of lectures is going to be in orange. So I talked about this at the end of chapter 17. All of your cardiovascular system stuff is in blue, and this set of lectures will be in orange. And I will continue to color coordinate as we go through the rest of the semester. So again, I'm going to be skipping over the chapter objectives, but feel free to read through these so you have an understanding of what we're going to cover in this lecture. So the growth structure of skeletal muscle will be the first topic. There's about 600 muscles in our body. They can range from the small muscles of our eyes that we use to move the eyeball around to the bigger muscles like our glutes and our quadriceps in our legs. And they contain various wrappings of fibrous connective tissue. Each muscle consists of thousands of cylindrical cells called fibers that lie parallel to each other with a force of action directed along the fiber's long axis. Now what this means is essentially it's a pulley system. So if you have something at the end of the pulley and you want to pull it up, that long axis or that string that's pulling that pulley upwards is going to be that long axis of the fiber. Fiber lengths can vary from a few millimeters in the eye to 30 centimeters in large leg muscles like we just talked about. So here's the skeletal muscle architecture. You guys should be pretty familiar with this, not only from anatomy, but also from your previous physiology courses. So this will kind of get you refamiliarized with some of the terminology, such as like the mitochondria, myofibrils, sarcolemma, and then kind of the greater breakdown of muscle fibers to a fasciculi, and then to the actual muscle itself. So here's the skeletal muscle levels of organization. Again, if you want to reference back to the pictures, this will give you guys a bit more insight, but I'll kind of read through each layer here. So the epimysium surrounds the entire muscle and blends into intramuscular tissue sheaths to form tendons. The perimysium surrounds a bundle of fibers and is called a fasciculus. The endomysium wraps each muscle fiber and separates it from its neighboring fibers. So you can see how these layers are basically, these mysiums are the surrounding tissue for each group of things. So the epimysium goes around the entire muscle, the perimysium goes around each fasciculus, and the endomysium goes around each fiber. The sarcolemma surrounds each muscle fiber and encloses the fiber's cellular content. The sarcoplasm contains nuclei that house the genes, mitochondria, and other specialized organelles. And the sarcoplasmic reticulum provides structural integrity to these smaller components. Next, we'll talk about tendons. So this just goes back into that levels of organization again. Tendons connect both ends of the muscle to the periosteum. So essentially, they're just saying that the tendon attaches to the outside of the bone. The muscle's force of action transmits directly from the connective tissue to the tendons, which then pulls on the bone at the point of attachment. So when you guys took anatomy, you learned about origins and insertions. Okay, so the origin is the location where the tendon joins to a relatively stable part of the skeleton, and then the insertion is the point of the distal muscle attachment, usually, of the moving bone. So if we look at your bicep, the origin would be up towards your glenoid, okay? And the insertion is on the radial head. So when you guys are making that muscle action of flexing your bicep, you're going to see that insertion move towards the origin generally. Now here's the muscle's chemical composition. So 75% of it is water, 20% of it is proteins. That includes myosin, actin, and tropomyosin, as well as some others. And then salts and other substances make up 5% of the muscle. So next we'll talk about blood supply to the skeletal muscle. So we have arteries and veins that lie parallel to the muscle fibers. So they're running in the same orientation as the muscle, and they're divided into numerous arterioles, capillaries, and venules to form a network in and out of the endomysium. Okay? Extensive branching ensures that adequate oxygen supply and rapid CO2 removal is possible. So you have a lot of these smaller vessels, especially these capillaries, that are allowing that oxygen exchange and that carbon dioxide exchange to happen at the level of the capillaries so that you can get oxygen to the tissues that need it and remove CO2 and bring that back to the heart so it can be sent to your lungs and then eventually breathed out. During intense exercise, the vascular beds deliver large quantities of blood through active tissues, and then physical activities that require straining elevate the intramuscular pressure to occlude local blood flow during muscular contractions. This is not as important as this. I think this is the biggest thing that we're trying to hit home with you guys is during exercise, you're starting to see this increase in blood flow so that tissues that need oxygen are getting them. Capillarization. So capillarization is a term that shows that over time, if you train a muscle, you're going to see more capillaries in that muscle. Your body gets to it gets to a point where it adapts to the stresses that are put on it. And as we steadily put more stress through a muscle, it learns that it needs more blood flow in order to do the work that it needs. So you start to see this increased capillary to muscle fiber ratio. This enhanced capillary microcirculation expedites removal of heat and metabolic byproducts from the active tissues and facilitates delivery of oxygen, nutrients, and hormones. So you have more capillaries, that means more oxygen and nutrients can get there. That means all the waste and carbon dioxide can move out. So in endurance athletes, we usually see a 40% higher volume of capillaries. So that makes sense. They're more trained. You're going to see more capillaries in that population. Now the skeletal muscle ultrastructure. So don't worry about the term so much, but we're going to talk about the pieces that go into the ultrastructure. So each muscle fiber contains myofibrils that lie parallel to the fiber's long axis. Myofibrils contain smaller subunits called myofilaments that lie parallel to the long axis of the myofibril. So you can see everything's just lying parallel to everything else, right? We have the muscle fiber, we have the vessels, and we have all of these myofibrils as well. The myofilaments consist of the actin and the myosin that account for about 85% of the myofibrillar complex. Other proteins either serve structural function or affect protein filament interactions during muscle action. So this could be tropomyosin, troponin, which those you're probably familiar with from previous courses. We got alpha-actinin, beta-actinin, M protein, and C protein. You don't need to know these four, but you will be familiar with these first two. So here's the skeletal muscle ultrastructure. We've already seen this. We know that this is a muscle fiber, and then we know we have these myofibrils in here, right? So if we break this down, we have our actin filaments, which are these thin blue ones, and then we have our myosin filaments, which are these thicker pink filaments with the arms on them. So the sarcomere itself. The sarcomere myofibril has a cross striation pattern. It has an I band, it has an A band, and that A band contains an H zone, and that M band bisects the H zone and consists of protein structures that support the arrangement of myosin filaments. The Z line bisects the I band and adheres to the sarcolemma to provide structural stability. If you zoom in to the photo in 2 slides from now, you'll see these structures. These are just good structures to familiarize yourself with so that you know what you're dealing with when you're looking at the sliding filament theory, which we'll talk about later in this chapter. So the sarcomere consists of basic repeating units between 2 Z lines. So this comprises the functional unit of a muscle fiber. So you're going to get all of these sarcomeres, a whole bunch of them in a row, and then that's going to end up being a muscle fiber. Sarcomeres line series and their filaments have parallel configuration with a given fiber. We've already talked about that. Everything lies parallel to itself. Sarcomere length determines the muscle's functional properties. So basically what this means is if the sarcomere is shorter or it has shortened, your muscle is going to be contracted or shorter. Or if the sarcomere has lengthened or stretched out, your muscle is going to be longer. So this kind of summarizes that point. So here's that visual representation I was talking about of a sarcomere. So you can see a sarcomere is from the I band to the I band. And the I band is essentially this area from the tip of a myosin filament to the tip of a myosin filament and encompasses this Z line. Right? So the Z line is actually right within that I band. And from I band to I band is a sarcomere. Within that sarcomere, we have the A band. And the A band is basically encompassing the pink, or these myosin filaments. Within that A band, there's an H zone. And that H zone is this area right here, where you're not getting that crossover of actin to myosin. So it's pretty much just this open area where there's not any pulling happening from the myosin heads. And then you also have this M band, and that's pretty much the attachment point where these two myosin filaments end up connecting together. And that pretty much summarizes these structures that make up a sarcomere. So the differences in sarcomere alignment and length affect muscles' force and power generating capacity. So there's two different types of muscle fibers. There's the fusiform muscle fibers, and there's pinnate muscle fibers. So pinnate muscle fibers differ from fusiform muscle fibers because pinnate muscle fibers contain shorter fibers, they possess more individual fibers, and they exhibit less range of motion. Now fusiform fibers, or spindle-shaped fibers, they run parallel to the muscle's long axis and taper at its tendinous attachment. So a great example of this is actually the bicep. So the bicep has these fusiform fibers. They travel down the length of your bicep or down the length of your humerus. And you can see like in your bicep, it gets thicker in the middle and then it kind of thins out towards the ends. So that's that tapering that happens at the end. And this parallel orientation allows for a facilitation of rapid muscle shortening. So because everything's lined up all the same, when you pull on that pulley, it can happen very quickly because there's a lot of fibers running in the same direction. On the other hand, we have pinnate fibers. So pinnate fibers actually lie at an oblique pinnation angle. So they kind of lie on these weird angles or these different angles that allow for the packing of large number of fibers into a smaller area. There's three different classifications of pinnate fibers. There's unipinnate, there's bipinnate, and there's multipinnate. The degree of pinnation directly impacts sarcomere fiber number per cross-section of area. So basically what this is saying is there's going to be less fibers in a unipinnate, there's going to be a few more fibers in a bipinnate, and there's going to be the most fibers in a multipinnate muscle. This degree of pinnation allows individual muscle fibers to remain short while overall muscle attains considerable length. So you don't have to lengthen these muscles significantly in order to create a lot of force. So here's a good picture of fusiform versus pinnate muscles. So I just talked about this. Here's your bicep muscle. It has a lot of linear force, right? So that pulley is just pulling up this way towards the shoulder, and that's what allows it to contract very quickly, is because all of the pulleys are pulling in one direction. The unipinnate, bipinnate, and multipinnate muscles, you can see that the orientation of these muscles kind of varies. There's different angles. These ones are kind of pulling up. This is your biceps femoris. This one looks like your tibialis posterior. It's kind of pulling up at an angle like this. And then this is kind of your shoulder complex, so your deltoid. So it's pulling a whole bunch of different directions. All of these fibers are oriented at different angles, and they have those oblique angles that are allowing a lot of muscle fiber to be packed in. So you could generate a lot of power without having to shorten that muscle a lot, right? So you can visibly see your bicep shorten. Like if you practice right now and you were to contract your bicep, you might actually see it shorten visually versus, your deltoid. It's pretty hard to see your deltoid shorten, but it still creates a lot of power. So just keep that in mind when you're looking at these two different types of muscles. So next we'll talk about fiber length and muscle length ratio. So the ratio of individual fibers length to a muscle's total length usually varies between 0.2 and 0.6. So the individual fibers in the longest muscles remain significantly shorter than the muscle's overall length. So this basically is trying to say, I don't care if you know the ratio itself. That doesn't matter to me. The thing that matters to me that you understand is that we get muscle shortening in the actual fiber, but the fibers themselves are shortening pretty significantly compared to the actual shortening of your muscle. So we're not getting everything just rolling up into a ball, because that wouldn't be good. We need to have a little bit of give in these muscles. So that's why when you contract your bicep, that bicep's not just gonna roll up into your shoulder and get so short, despite the fact that that the muscle fibers are shortening fairly significantly. So here we'll kind of talk about architectural properties, and I think we're going to focus more on this graph on the right-hand side here. And what you're going to see in this graph is based on muscle length, you're going to see a certain amount of force. So typically, when you have a bit of a shorter muscle, you're going to generate more force. When you have a longer muscle, you're going to generate less force. And then the same is true with muscle velocity. The faster it's moving, the more force you can create, right? It's like this muscle, for example, created a lot of force and it moved very quickly and this one moved a little bit slower. It was going 200 millimeters per second and it didn't create as much force. So those are things that you want to keep in mind is, speed and velocity can influence the force, and then the actual length of the muscle itself can change the force. So next we're going to break down the sarcomere a little bit more, and we're going to talk about the sliding filament theory. So we're going to start with the actin-myosin orientation. So we have thousands of myosin filaments that lie along the line of actin filaments in a muscle fiber. These myosin or thick filaments are bundles of molecules with polypeptide tails and globular heads. You guys have seen this. This is the pink one with the little tails that come off of it. And then you have the actin or thin filaments that have two twisted monomer chains bound by tropomyosin polypeptide chains. And this is the thin blue one that gets influenced by calcium and troponin. But for simplicity's sake, just remember it as the blue line that myosin attaches to. 6 actin filaments encircle one thicker myosin filament. So we have lots of actin surrounding one myosin. In a single muscle fiber, we actually have about 16 billion thick filaments and 64 billion thin filaments. So this is just a fun fact for you, but you can see that there's just an enormous amount of filaments that exist within one single muscle fiber. So here's another visual representation for you guys. We talked about this A band, the H zone, the M line would be here, you got your Z line. These are all things that you guys should be aware of from previous courses, so I won't spend too much time here. So cross bridges, you guys are probably familiar with this term too. This is when the myosin head extends across and latches onto the actin strand. And this creates a structural and functional link between myofilaments. ATP hydrolysis, so the breakdown of ATP, activates the myosin's two heads, and places them in optimal orientation to bind to the actin's active sites. The myosin then pulls the thin filaments and the Z lines of the sarcomere towards the middle. So the Z lines get pulled together. Tropomyosin and troponin regulate the make and break contacts between the myofilaments during muscle action. We're going to talk about this later, but this is kind of where calcium comes into play, and it's going to influence if those sites on the actin filament are available to the myosin or not. So here you guys go. There's the actin filament in blue. There's your myosin filament in pink. You got your myosin heads coming up. This is your tropomyosin is going to be kind of your attachment point, and this troponin complex is also involved. So just another visual for you. So the intracellular tubule systems. So this kind of relates back to that sarcoplasmic reticulum. So the lateral end of the tubule channel within muscle fibers terminates in a sac-like vesicle that stores calcium. The T-tubule system runs perpendicular to a myofibril, and it functions as a microtransportation network by spreading action potential from outer membranes. So you get this action potential, it comes down the T-tubule, and then it triggers that T-tubule to release calcium and diffuse a short distance to activate the actin filaments. So you have this calcium rushing in from the T-tubule and then activating these actin filaments. So muscle action begins when the myosin filament cross bridges or attaches to the active sites of actin. The electrical excitation stops, calcium concentration decreases, and the muscle relaxes. So after all of that process has occurred, you get this cessation of electrical excitation, you get this decrease in calcium, and then you get the muscle relaxing. So here's another visual for you guys. So we have this, this neural excitation that comes to the T-tubule. This T-tubule releases calcium into the sarcomere, and that sarcomere accepts the calcium in that tropomyosin and troponin complex, which allows the actin to have those active sites available for the myosin. So this is a very general drawing for you, but hopefully makes it a little bit easier to follow. So next we have the chemical and mechanical events during muscle action and relaxation. So this is where we're going to talk about the sliding filament model or the sliding filament theory. It was proposed a long time ago and has undergone a few changes, but it remains fundamentally the same. So let's start to talk about that now. So the sliding filament model proposes that a muscle shortens or lengthens because of the thick and thin filaments sliding past each other without changing length. So the myosin attaches, rotates, and then detaches from the actin with the energy of ATP hydrolysis. This produces a change in relative size within a sarcomere zones and bands and produces a force at the Z bands or the Z lines. Those Z lines get pulled together. The I band decreases as the Z lines are pulled towards the center of the sarcomere. So if you guys go back and look at the I band, you can see where the I band starts to decrease over time because those Z lines are getting yanked together by the myosin heads continually attaching to those actin sites and pulling it closer together. In an isometric muscle contraction, the fiber's length remains unchanged, so relative spacing of the I band and A band remains constant. So an isometric contraction, as you guys I'm sure know by now, is when the muscle actually doesn't change length. So a great example of this is when you're carrying a box. Your biceps are working, you're having to contract, but the muscle is actually not shortening. Like you're not raising that box up towards your face, you're keeping that box there. So in this example, the muscle fiber length stays the same and the I band doesn't change. So you're still getting this attachment of the myosin to the actin without it actually pulling those Z lines together. And if you guys don't like how I explain this in words, you can also watch this video on the sliding filament model. So you should have this in your slideshow that you can just click on and go take a look at that. So here's rearrangement of the actin and myosin during muscle shortening. So here is at rest. We don't have a lot of cross bridging between the myosins and the actins at rest. And then as we start to contract and we start to shorten that muscle, we start to see this thick filament attached to the thin filament, and these Z lines you can see get pulled closer and closer together until they have a full crossover like this. Now we'll talk about the mechanical action of cross bridges. So myosin plays both an enzymatic and structural role in muscle action. The globular head cross bridges, which provides mechanical power stroke for the actin and myosin to slide past each other. So it's basically just acting like an oar. So as you put that oar into the water, it's allowing you to pull through the water, and that's what that action of the myosin is like. This will continue to happen, so it'll be cyclic and oscillating to and fro, which is powered by ATP hydrolysis. So as the ATP breaks down, you continually have this oar-like action on the actin, which allows those Z lines to be pulled together and the muscle to shorten. So here's some actin and myosin oscillation. Again, it's just showing you how they cross bridge, and then it gets pulled, and then the movement continues down the line of the actin. So here's the sarcomere length isometric tension curve in isolated fiber. So we're just going to look at one single muscle fiber here. And we're going to look at the sarcomere length in relation to isometric tension. Well, if there is less overlap in the muscle, like let's say your muscle is quite elongated, you're going to have less tension, right? So you're not going to be able to produce as much isometric tension when you have a lengthened muscle. The opposite is true though. If you have a more shortened muscle, then you're going to be able to create more isometric tension generally, right? Like there's a balance to it. You can't have it super, super short, or else you kind of lose a little bit of power there. But you're going to have that midpoint where you're going to have that perfect amount of crossover between actin and myosin that's going to create the most amount of isometric force. So big thing here, kind of looking at this. The less overlap, the less tension. The more overlap, the more tension, but there's a balance to those two things. And here is that balance. So here's the interaction between actin and myosin during an isometric contraction. So if it's too short, you get too much crossover here, well, you can see your maximum tension starts to go down pretty significantly. There's almost too much crossover, right? You're not getting that optimal amount that you need to create the most tension possible. However, when it's too long, it's the opposite is true. You're getting almost no crossover of the actin and myosin, so you're definitely not optimal, so you're not going to create as much tension here either. So you start to see these drop off on each end. When you have that perfect crossover of the most myosin heads attaching to the most amount of actin, then you are going to get this optimal sarcomere length that's going to create the most tension with an isometric contraction. Here's just another visual. It's just actually showing muscle fiber here. You don't need to worry about that too much. Just basically showing the same thing from last slide. So here's the link between actin, myosin, and ATP. So the myosin cross bridges detach from the actin filament when ATP molecules join the actomyosin complex. Myosin cross bridges in this chemical reaction return to its original state, ready to bind to a new active actin site. So the dissociation of actomyosin occurs as follows. So before we get into the actual formula here, just know that actomyosin is when the myosin head is attached to the actin site. So we need to be able to dissociate these two things. These two things need to be able to pop apart so that you can get the next myosin head onto the next actin site so you can continue to shorten the muscle. So when we have actomyosin or actin attached to myosin and we add ATP, eventually we're going to pull apart these two parts, you're going to have actin separated from myosin, and myosin is going to have ATP kind of connected to it. Okay, and this is going to get it ready to bind to the new active actin site. Finally, we'll see myosin ATPase split. So ATPase, that's just a enzyme that breaks down ATP to yield energy for the muscle action. So once it gets broken down, it attaches back onto an actin site. It makes that oar-like movement, and we start moving those Z lines together. Next is excitation contraction coupling. So this represents the physiologic mechanism whereby an electrical discharge at the muscle initiates chemical events at the cell surface to release calcium into the sarcomere to produce muscle action. So we talked about this earlier. When we get that neurological excitation, it's going to excite the T-tubule and it's going to release calcium out into the sarcomere so that the calcium can regulate the muscle fiber's contractile and metabolic activity. So essentially what calcium is doing is it's freeing up the actin sites so that myosin can attach to it. So don't worry about these formulas too much and just focus on the process of how this all happens. So here's a great visual for the last slide. So here you have your myosin filament, you have your actin filament, and then you have your troponin complex and your tropomyosin and your calcium binding sites. So right now the myosin can't attach to the actin because tropomyosin is blocking, this tropomyosin filament is blocking these troponin attachment sites or these actin attachment sites for the myosin heads. So how does that get released? Well, it's actually because of calcium. So when that signal comes down through the T-tubule and the calcium gets released into the sarcomere, all of a sudden these calcium ions will start to attach onto these calcium binding sites. And they will actually free up the tropomyosin and allow the myosin heads to attach to the actin. So this is where calcium is very important in muscle contraction. Now, that's muscle contraction, so let's talk about relaxation. This occurs when actin and myosin return to their original states. So when the muscle stimulation stops, the calcium activity stops and troponin frees up to inhibit actin-myosin interaction. So recovery involves active pumping of the calcium into the sarcoplasmic reticulum. So now we have to get all the calcium out of the sarcomere, right? So the deactivation serves two purposes. One, it prevents any mechanical link between myosin cross bridges and active filaments. So we are detaching all of the myosin from the actin. And then two, it inhibits myosin ATPase activity to curtail ATP splitting. So essentially what this means is you're stopping the enzyme that breaks down ATP to continue to break down ATP, right? You want that ATP energy release to stop so that you're not continuing to attach myosin onto actin over and over and over when you're trying to relax your muscle. So here's the sequence of events in muscle action. So I've already talked about this quite a bit. So this is going to be a bit of a review, but hopefully this gives you a bit of a step-by-step if you're having a hard time following. So step one, we have an action potential in a motor neuron that causes terminal axon to release acetylcholine, which diffuses across the synaptic cleft and attaches to a specialized acetylcholine receptor on the sarcolemma. Step two, the muscle action potential depolarizes the transverse tubules at the sarcomere's AI junction. So these are the T-tubules, right? We talked about the T-tubules getting excited. Then what happens? Step three, the depolarization of the T-tubule system causes calcium release from the lateral sacs of the sarcoplasmic reticulum. So now you have calcium getting released out into the sarcomere. Step four, the calcium binds to the troponin tropomyosin in the actin filaments releasing the inhibition that prevented actin from combining with myosin. So we just talked about that. Calcium attaches to those calcium binding sites, but troponin tropomyosin complex gets moved out of the way so that step 5, the actin can combine with the myosin ATP, which activates myosin ATPase, which splits ATP. So now the myosin can attach to the actin and the ATP can break down. This reaction's energy produces myosin cross-bridge movement and creates tension. So now you have this energy that gets released and these two pieces are connected together, the actin and the myosin, which causes muscle shortening. Step 6, the ATP binds to the myosin cross-bridge, which breaks actin-myosin bond, allowing actin dissociation from the cross-bridge. So now this ATP is coming in and breaking those two pieces apart, and then the thick and the thin filaments slide past each other and the muscle shortens. Okay, so this is that pulling action. Step 6 is where those Z lines get pulled together from that oar-like action as you're pulling through the water. That's step 6. Step 7, cross-bridge activation continues when calcium concentration remains high enough to inhibit the troponin tropomyosin system. So as long as we have calcium in the system and calcium in the sarcomere, there is going to continue to be shortening that happens as long as that's the neural input that's happening. Okay, so as long as there's calcium there, it's going to continue to shorten. Step 8, when the muscle stimulation ceases, so when we stop getting that neural input, the intracellular calcium concentration rapidly decreases and calcium moves back into the lateral sacs of the sarcoplasmic reticulum through active transport, which requires ATP hydrolysis. So you still have, we talked about this earlier, you have all this calcium in the sarcomere. Now muscle activation has stopped. So now I need to get all this calcium out. So I need to actively transport it out, which I need ATP for. It breaks down, the calcium gets taken out of the sarcomere, and then we're on to step 9. Calcium removal restores inhibitory action of troponin tropomyosin, and in the presence of ATP, actin and myosin remain in a dissociated, relaxed state. So now you've removed all the calcium, troponin and tropomyosin will now attach back onto those spots so that the myosin heads can't attach, and now the actin and myosin are separated from each other. So we just talked about these 9 steps, but here it is in picture form. So if you didn't like the words, here's a visual like for you. You can always reference back to this. I personally like the pictures. Makes it a bit clearer for me, so feel free to use this as a reference. Next, we're going to talk about muscle fiber types. Okay, so the skeletal muscle contains 2 main types of fibers that differ in 3 characteristics. There's primary mechanisms used to produce ATP, there's the type of motor neuron innervation, and there's the type of myosin heavy chain expressed. The proportions of each type of muscle fiber vary from muscle to muscle and from person to person. Now, you're probably wondering what this slide means. Well, don't worry, we're going to get to that. So now you're probably looking at this and you're saying, okay, I recognize what muscle fiber types are, right? We have your type 1, we have your type 2A, we have your type 2X, we have your type 2B fibers. Now, these have been changed since I've been in school even. We have this this addition of these type 2B fibers. So that's been added in since I've gone to school. And with research, we start to discover these new things. But the things that I want you guys to be aware of are the things in bold, right? It's not that this stuff down here isn't important in the future, but for right now, for what you guys need to know, I think this is the things that you want to focus on, right? What's the contraction time? What's the size of the motor neuron? What's the resistance to fatigue? What's the activity used for? What's the maximum duration of use? And then what's the force production? And then another thing you guys can think about is, what kind of sports or what kind of activities are type 1 fiber biased? What kind of activities are type 2 fiber, right? So really think about these things, and that can help you if you do want to go into the health sciences. You want to be able to train athletes to be specific to their sport. Now, you're not going to be able to necessarily control what type of fibers they make, but just knowing that some of these, some of these things here, that can help you train that person to bias towards a particular fiber type. So we'll talk about fast twitch fibers first. These are the type 2s. They exhibit 4 characteristics: high capability for electrochemical transmission, high myosin ATPase activity, rapid calcium release and uptake by efficient sarcoplasmic reticulum, and a high rate of cross-bridge turnover. So this just means that they just move fast, right? So they can get those myosin heads attached and released way faster than other fibers. So these factors contribute to this fiber's rapid energy generation for quick and powerful muscle actions. Now, fast twitch fibers, their intrinsic speed of shortening and tension development ranges from 3 to 5 times faster than slow twitch fibers. They rely on well-developed short-term glycolytic system for energy transfer, and activation predominates in anaerobic type sprint activities and other forceful muscle actions that rely entirely on anaerobic energy transfer. Activation plays an important role in stop and go or change of pace sports such as basketball, soccer, lacrosse, field hockey, ice hockey, anything like that. You want to have that really rapid response that allows you to change pace quickly or change direction quickly. Type 2 fibers are distributed in 3 primary subtypes. You have your type 2A, which is your fast oxidative glycolytic, or your FOG fibers. You have your type 2X, which used to be called type 2B, which is the midway point between type A and type B. And then you have your type 2B, which is your greatest anaerobic potential and your most rapid shortening velocity, which represents true fast glycolytic fibers. These are the fastest ones. These ones are going to contract the quickest, then these ones, and then these ones will be the slowest of the type 2. If you go back to that table I showed you and those bold important things that I wanted you to know, you'll be able to reference back and make a differentiation between these 3. Now, slow twitch fibers. These are type 1 fibers. They generate energy for ATP resynthesis through the aerobic system. So lots of anaerobic with the type 2, lots of aerobic system utilization in type 1. There's 4 distinguishing characteristics of slow twitch fibers. 1, low myosin ATPase activity. 2, slow calcium handling ability and shortening speed. 3, less well-developed glycolytic capacity than fast twitch fibers. And 4, large and numerous mitochondria. So slow twitch fibers are highly fatigue resistant, so they don't get tired very quickly. So they're ideally suited for prolonged aerobic exercise. So these are like marathon runners, triathletes. These people have a huge range of slow twitch fibers in their system. These fibers are slow oxidative fibers with slow shortening speed, and they rely on oxidative metabolism. So we're not relying on glycolytic metabolism as much as we're relying on oxidative metabolism. Both the slow and the fast twitch muscle fiber types contribute during near maximum aerobic and anaerobic exercise. So we sometimes, I know when I went through school, I thought that aerobic athletes like marathon runners, they were just all type 1. Well, it's not true. We actually have a mix. So even for elite athletes that train at very, very high levels, on one end or the other, they actually have a mix. They might have more of one that they've trained for, but they're always going to have a mix of different muscle fibers. So how do we find out what kind of muscle fibers you have? Well, we take a muscle biopsy. So they actually go in there, they'll cut a piece of muscle out, and then they can actually analyze what your muscle fiber type is. So here we go. Here's your unstained ones. Here's particular pHs, right? So when you stain the muscle fibers, you can actually pick up which ones are which just based off the staining and the acidity of that staining. I personally have never done this, but kind of an interesting experiment that allows you to pick up on different muscle fiber types through a biopsy. So there are fiber type differences among athletic groups. I've kind of talked about this. You're going to have your marathon runners that are going to have a bit more type 1 fibers. You're going to have your sprinters or really speed athletes that are going to probably be more biased towards type 2. But here are some general trends that we kind of see generally among the population. So men, women, and children on average possess 45 to 55 slow twitch or percent of slow twitch fibers in their arm and leg muscles. When you do have fast twitch fibers, they distribute equally between type 2A and type 2X subdivisions. So you end up kind of getting this blend of 2A and 2X when you do have those fast twitch fibers. Although there are no gender differences that exist in fiber distribution, large inter-individual variation occurs. So, males aren't going to have more type 2 fibers than females. That's not an actual physiological correlation between sex and fiber type. But if you train for a particular activity, like if you are a marathon runner, like I said, you're going to have more type 1 versus a sprinter who is going to have more type 2. And finally, a trend in one muscle fiber type distribution remains fairly consistent among the body's major muscle groups. So if you are a sprinter and you train and you have more type 2 muscle fibers, your body's going to adapt to that kind of consistently throughout your body. So you're not going to get these huge differences between your arms and your legs, for example. You're going to get that change throughout your entire system. So next, we've already talked about this, but endurance athletes have more slow twitch fibers and elite sprint athletes have more fast twitch fibers. Now, the performance success or the success of an athlete doesn't just depend on muscle fiber composition. So just because you have a bunch of type 1 muscle fibers and you're a marathoner doesn't mean you're going to be successful. There's a lot of other components that go into it. There's physiological reasons, there's biochemical reasons, there's neurologic reasons, there's biomechanical reasons, and some are arguing now there's psychological reasons, right? So there's other drivers that can help you have success as an athlete. Larger muscle fibers in male athletes, along with larger total muscle mass, are the principal gender differences in muscle morphology between males and females. So if you have a male sprinter and a female sprinter, basically the biggest difference between those two is going to be the fact that the male's going to be slightly larger because he can carry more muscle mass and those muscle fibers are going to be a bit bigger. Aside from that, the muscle fiber composition will be very, very similar. They're going to have a similar, if you were to take a muscle biopsy, they're going to present pretty similarly. So the big difference between males and females at that comparison is going to be the actual size of the male athlete and their muscle mass. So here's fiber composition and VO2 max in athletes. Not really something you need to know, but kind of a cool graph, just a cool graph to show you guys what the muscle fiber composition is in different athletes and that relation to their VO2 max. So you can see cross-country skiing, they have a bunch of slow twitch fibers because they're doing lots of aerobic work. Their VO2 max is super high. You can see down lower, you got the sprint athletes. They don't have as many slow twitch fibers, and their VO2 max maybe isn't quite as high because they don't need it to be. They're just sprinting super quickly. So they don't need slow twitch fibers. They don't need to be super aerobic athletes. They need to be anaerobic athletes that can run super fast and have lots of fast twitch fibers. And that's it. That's chapter 18. So here's the summary. Feel free to read through this. Again, it will just summarize everything that we've talked about. So if you guys have any questions, don't be afraid to reach out. Again, I have put this in orange for you guys for these couple chapters that summarize the muscular system. And if you have any questions at all, don't be afraid to reach out, and we'll see you for chapter 19.